I just want to thank the organizers. It's great to be back at 3DSIG. I think I've been to many of these meetings. So what I'm going to talk about today is using density-based clustering methods in structural bioinformatics. And this is work by graduate student Simon Kilo and a senior scientist in my lab, Maxim Shapovalov. So about 7 years ago, we published a clustering of the CDRs in antibodies. And this had been done by Chothia in the 1980s, but had not been done in many years, and by Jenna Thornton also, in many years. And so we had a lot more data. We thought we would redo it. And so we came up with a kind of standardized nomenclature for the CDR conformations. So H2 is the CDR2 of the heavy chain, length 10. The biggest cluster is H2-10-1. The second biggest one is H2-10-2, and H2-12-1 for length 12. And this nomenclature is now being used, I think, by a lot of groups. When you see an antibody structure paper, sometimes they're using our clustering. But we want to be able to update it, and one of the issues that came up over the years is outliers, things that are not near any of our clusters, or it don't seem to be like other structures in the PDB. One thing as an aside, we have a website called PyAG Classify, where you can put in any PDB code, or you can upload your own structure, and it will tell you what clusters there are, the sequences of the CDRs, the germlines, et cetera. And you can click on one of these cluster identifiers and get all of the members of that cluster within the PDB at any one time. But over the years, we've started to use a nomenclature, something like L1-11 star, which means a structure of a CDR of length 11 that's not near any one of our cluster centers. And as time goes by, the size of those starred clusters has gotten larger. So clustering is used in many areas of structural bioinformatics. And there are certain kinds of clustering situations that are more complicated. So on the left, we have 3 different clusters. They're Gaussian distributed. They're very easy. On the right, these are a little bit too close together. And exactly, by eye, you can see where the clusters are. But a clustering algorithm might be confused by them. So the clusters can have different densities, and they can have different shapes. They can even have weirder shapes, where, again, your eye tells you what the clusters are. But if you used a simple clustering like k-means, you would get not the answer that your eye would pick out. And what's probably even more relevant to structural bioinformatics is we have a lot of noise. We have structures in the PDB that are wrong. If you want an interesting story, I can tell you the structure of an antibody in the PDB that is probably fraudulent. Or structures that are poorly determined, low resolution, or even just weird because somebody put in, say, a random sequence for a CDR that's not related to any of the germlines, and it's going to look different than every other CDR. Maybe an important structure, but it's not going to fit into one of the clusters. And so we have a situation like this where we have very observable clusters, but we have noise points. Now, there are lots of standard clustering algorithms. I'm not going to go through all of them. K-means or K-medoids, just you give it a number K, how many clusters do you want, and it starts randomly, picks out 3 different centers, and then assigns everybody to their closest center. Then with those 3 clusters, it picks out a new center and just repeats that process over and over again. On the right, we see hierarchical clustering. This means we start with everything in its own little cluster. You unite the closest ones, and then start uniting the groups. Again, that's a very standard algorithm. An algorithm that I was less familiar with, as we're sort of starting to look at, is there a different way of doing this clustering, are density-based clustering methods. And so on the left, we see a bunch of clusters that, again, your eye easily picks out. We also see noise points. There are algorithms called DBSCAN, DEN-CLU, there's a whole bunch of them, that while they're doing the clustering, they are looking at the density, and they're looking for regions of high density separated by regions of low density, and the regions of high density become the clusters, the regions of very low density might become the noise in between the clusters. And so you're really looking, again, for what your eye actually sees, which is high density, low density, high density again. Those are the clusters. So the algorithm that we're going to use is called DBSCAN. It comes from 1996 from Esther, Kriegel, Sander, and Shu. And the idea behind DBSCAN is you have 2 different parameters. One is called min points, and one is called epsilon. A core point, sort of sub-points that are going to end up in the clusters, we'll call them core points, are core points if they have at least min points within epsilon. So here's a core point. Min points is 4. Epsilon is this circle. There are at least 4 points. There's actually 5 or 6 here. Points around that within epsilon. So we're going to call that a core point. A point that is not a core point, but is within epsilon of something that is already designated a core point, that's going to become a border point. And everything else is noise. And so the way the clustering works is once you've identified what's a core point, what's a border point, what's a noise point, you sort of treat it like a graph. All points that are within epsilon of each other, all core points that are within epsilon of each other become a cluster. Those border points get added to their nearest set of core points, and the noise points remain noise. You can think of min points when you sort of use this in practice. Min points is the size of the smallest cluster desired. The smallest cluster you're going to get is going to be roughly the size of min points. As long as there are sort of min points within epsilon of each other, that's the sort of cluster you're going to get. That's the minimum density. So if we apply this to that same data set that I just showed, on the left, this is what happens with k-means. These sort of centroids that you get of the centers are kind of displaced from the actual centers, and the reason is that the noise around them is not quite uniformly distributed. But on the right, all of those points have now become noise, and so now the centroids really are closer to the actual centroids that I use to generate these data, to sort of a random Gaussian distribution. So we're going to use DBSCAN for the CDR data. The way we're doing this now is that we're using redundant data. We're using every CDR of L1, length 11, no matter it's the same sequence as other CDRs, whether it's low resolution, high resolution, everything. What we hope, and what we're sort of still analyzing, is that structures that are incorrect or low resolution or for some reason wrong will end up in the noise points. Anything that ends up in one of our clusters probably belongs there. We're going to search a grid of min points and epsilon and sort of try to figure out what's the best combination of those two variables. This is true in every kind of clustering algorithm, that you have to kind of play around with your parameters until you get kind of what you're looking for. I've already said that. We're using what's called an L-infinity metric, which means in this case we're looking at the dihedrals phi, psi, and omega over 11 residues of L1, length 11. And we're taking the biggest, so between the distance between one loop and another one is going to be the biggest dihedral angle difference. And it's actually a function of that, which is 2 times 1 minus cosine of that delta theta. So we're looking for the biggest difference in dihedral angles. You can use the average distance or the total distance as well. It works pretty much the same way. This is pretty robust with the min points and epsilon. It's easier to choose min points and epsilon with this. And we're going to choose those values so that conformations within the clusters are uniform. I want kind of the same thing, things in the same region of the Ramachandra map to be grouped together. And I don't want things on the left-hand side of the Rama map to be paired with things on the right-hand side in the same cluster at the same residue, for instance. And then we're trying to minimize the number of noise points. I don't want just tight clusters of points and have 50% of the data go into the noise. That would be useless. Oh, and then finally, once we get the clustering, as I said, we're using redundant data. We might get some clusters that have all exactly the same sequence, maybe 10 structures of one antibody that were determined by one group, and it's the same sequence. I don't want that to be a cluster in our updated nomenclature, so we're going to filter those out. So if we take L3, which is CDR3 of the light chain, length 8, and we apply DBSCAN to it, these are the clusters that we get. So every row is a cluster. The bottom row here are the noise points. These are Ramachandran maps, so phi by psi, which I'm sure all of you are familiar with. So we have 8 residues, so we have 8 of those. Those are the noise points. And you can see some of them, a lot of the noise points are in regions of the Ramachandran map that are usually not that highly occupied, so it's not surprising that those are in the noise. The next one up is the biggest cluster. That corresponds to L381, which was a cluster in our work from 6 years ago. But this cluster in red is actually new. It has a different conformation if we label the different regions of the Ramachandran map. And the interesting thing is that at position 7, it very strongly has a proline. And so there's a new set of structures that have come out in the last 7 years that this is sort of a new cluster. If we look a little bit further at those structures, so here's L381. There are 27 unique sequences in there. This is our old L382. 7 years ago, it only had 3 sequences. It still has only 3 sequences. This did not become a kind of long-term cluster in the history of antibody structures. Maybe it will, but not yet. But L383, this new cluster, has 20 sequences. And then here are all the noise points. my time here. If we look at length 11, L111 is my favorite CDR length. These are the old clusters. We had 3 of them. Now we have 5, and the numbers are significantly increased. One thing that we're looking at is, are the members of each cluster kind of in the same region of the Ramachandra map? So if we break up the Ramachandra map into 4 regions, just this kind of low resolution alphabet of A, B, L, and E, this number % Rama is the percentage of members of the cluster that have the same string, that have the predominant string. And so back in 2011, this very small cluster was 5 members. Only 2 of them really had one Rama map, and the other 3 had different Rama maps, different Rama strings. Whereas that cluster now, there's 98% of them have that, and we have 23 members. So that's a feature to look for when we're doing this clustering. Do we get kind of uniform Ramachandra distributions? We've also applied this to the DE loop in antibodies. So antibodies have the 3 CDRs on each chain. There's also another loop that is adjacent to L1 and to H1 that's sometimes called L4 or H4 or the DE loop because it connects strands D and E. So we did clustering on that. This is just the light chain ones, which are length 6, both in kappa and lambda generally. And the noise cluster is down here. The biggest cluster are all the kappa CDRs. And then right above it is a cluster that is mostly lambda with a few kappas thrown in. And the difference between, one big difference between kappa and lambda is that the lambda first residue is usually not glycine. It's usually arginine or lysine. And so that flip actually changes that first amino acid. And occasionally when kappa has an arginine or a lysine, sometimes from the germline, it also changes to that second conformation. They're related to the conformation of that DE loop is strongly related to the conformation of L1 because they sort of intimately interact with each other. So in magenta is the DE loop of lambda chains and its CDR. In blue is the DE loop of the kappa chain and its CDR. And so there's a sort of complementary motion of those 2 structures. Now we're applying this to beta turns. This goes back to the 70s when beta turns were analyzed for the first time in protein structures as the early structures of proteins came out. And there's an old nomenclature, type 1, type 2, 1', 2', et cetera, that comes from that time period. So we applied DBSCAN to a very high resolution set. One of the reasons for doing this again and revisiting this problem, it's a very old problem, is because now we can do this with a 1.2 angstrom resolution data set. So this is over 1,000 chains, 13,000 beta turns from 1.2 angstrom crystal structures or better. So if we just run DBSCAN on that, we get 11 different clusters. They're sort of colored here. They're all overlapped, sort of hard to see. 2.3% of the data became noise. So that just removed a lot of the points that we think might be harder to cluster, and we're just going to remove those. Maybe we'll reclassify those later. We got back some of the beta turns that you would expect to get back, many of them. So this is the type 1 prime, left-handed, glycine at that position. This is a type 2 prime. This lower corner really needs to be a glycine, so it is, and an alpha helical region. And these are the sequence logos that you get out of those. We got some clusters that at one point had names, but no longer do, or at least those names are no longer used. So if you're familiar with beta turns, there's one type 1, 1', 2, 2', type 8, 6A, 6B are the cis ones, and type 4 are all the generic ones. But there is an older nomenclature. There are old turn types that have names. So this one at the bottom has a sort of alpha region over here and an alpha left region over here. And in the old days, that was called type 5. But that name hasn't been used in a long time. But you can find it in Jane Richardson's Anatomy and Taxonomy of Proteins, so that's where I found it. And it has a glycine at that position. There's also this one, which is this lower right-hand corner, and then sort of a beta conformation up there. And that one, I don't know that that one ever had a distinct name. Some of the clusters we got were too large. So this is the type 1 and type 8. So type 1 are alpha, alpha, and type 8, in the old nomenclature, is alpha and beta, sort of up here. But instead, DBSCAN was completely unable to separate those into separate piles. And so what we did is we applied k-medoids to the resulting points from this sort of set of data. And just by eye, picked out 4 different clusters. And these are the different sort of sequence logos for these different 4 clusters. This one in particular has a proline at position 4. This is the Raman map of residue 3. And so this is the pre-pro kind of conformation of residues at this position of residue 3, because there's a proline there. That conformation is allowed or favored when proline is there. So we're sort of breaking things up into smaller groups. So we started with 11, and by the time we broke up ones that looked like they had multiple peaks, that multiple things that should have different names or different piles, we came up with 17. We might narrow this further. I think 17 might be a couple too many, but this is what we have. Andy Karplus a few years ago did a lot of analysis in the Ramachandran map and started assigning labels to the entire map, and a bit more detailed than the ones that we typically use. So we're breaking up the alpha region into alpha and delta. And then I'm using small letters for the right-hand side of the map. So if we have capital A and D on this side, we have lowercase a and d on this side. B and P up here, we're used to. This corner, B and P, lowercase down there. And if we do that, we get this sort of list of clusters. And so I've got the new nomenclature and the old one. AD is type 1, lowercase ad is 1 prime. These are the type 8 that we've now broken up into 3. Type 2, it's inverse. These are sort of new clusters. This would be the type 5, this would be the type 5 prime. Again, those names have not been used in a long time, so on some level, they're new clusters. And then we found the cis3 ones and a couple of ones where the second residue was a cis, and those have never had names as far as I'm aware. We can look at the frequencies, and I tell my students that almost anything we do, Janet Thornton has looked at it before, sometime in the last 40 years. And of course, she did something very smart and elegant and thorough. And so the paper to go back and look at in this case is a paper that Janet published in 1991, which I think the title is A New Nomenclature of Beta Turns, that for whatever reason didn't take off. I think possibly because it had Greek letters and subscripts. And people just, who wants to typeset that? So this is very similar, but without the Greek letters and without the subscripts. But what we have now is a more quantitative, because we have a much bigger data set, we can be more quantitative. Both the frequencies and the phis and psis and their mean values and their variances and all sorts of properties and the sequence, the sequence information that we can do on this beta turn set that really wasn't possible more than 25 years ago. I'm not going to compare all the numbers, that's too much detail. And then finally, to bring both parts of the talk back together, is we're now looking at our CDRs in terms of what beta turns they contain with the new nomenclature. And so this is L1, length 11, cluster 1, and it has 2 beta turns of those 4 residues and those 4 residues. So this table just has all of our different L1, 11 clusters and the different kinds of beta turns they have. And you can see that each one of them has 2 beta turns at, in some cases, 95% of the examples in our cluster have the same beta turn type at the same location. And of course, the beta turn types are different in each one. So just to conclude, we can now use really high resolution data sets. For the turns, 1.2 angstrom. The density-based clustering removes noise. It helps us define real clusters in a way that k-means does not. There are more advanced methods that can handle different clusters of different density, which DBSCAN has some trouble with. And of course, we can all use all of this for protein structure prediction and design. So the work was really done by Maxim Shapovalov, who was here, and Simon Kilo, who was here, and this is the rest of the group. Thank you.
Hello?
Yes.
Hello. Thank you very much. So the question is, what metrics did you use to assess the quality of the clusterization?
We use something called a silhouette score, which for every point you see like what's the average distance of that point to everything in its own cluster, and the average of that distance of that point to everything in the nearest cluster. And it's a difference based on those two things. Then you average it over all of the different, sum it up for all the points in your data set. We did a lot of it by eye, but the silhouette score is sort of the quantitative thing that we're using to try to figure out whether one clustering is better than another.
And just one more question. Would you recommend using multiple metrics to assess the quality of the clusterization? So maybe the Davis-Baldwin index with silhouette analysis or a different combination?
Yes, I think so. I mean, I think it's definitely worth being quantitative. There is still no substitute for your own scientific judgment of what clustering is useful for the purpose that I want it. It's sort of, I think of it in kind of a Bayesian way of a prior distribution over the clusters that seem reasonable and useful given what I know about protein structure. But yes, I agree with you.
Thank you very much.
Last question.
Very nice, beautiful.
Oh, thank you, Janet.
But when we were doing the turns, the real thing was to try and look at loops and see to what extent they constrain or were constrained by the bits that came in. So the question is, are you taking this, which is the sort of turny bit, and then you've done it for the CDRs, but are you going to do it across the whole? And do you find that any other loop constraints that are bigger than just the turns?
Yes, absolutely, and thank you for the question. In fact, one reason this came about was doing loop structure prediction over the years and looking at long loops. If you look at loops that are longer than 8 residues, about 50% of the residues are in beta turns. And so, you know, loops are not like this. They're, as you know, they're sort of windy like this. They have a lot of internal structure. And so I wanted to get at the beta turn question and to put that in an algorithm in Rosetta or in any other program. Does Rosetta sample beta turns sufficiently? Does it sample them at the right rate? Does it score them at the right rate? And then what other kinds of constraints can we put on it? You know, can we figure out, well, at this sequence we should be testing type 1 turns 50% of the time and some of the other turn types. So it's a way of sampling in structure prediction that we would like to use it in loops in any kind of protein.
Okay, let's thank Roland for the nice presentation again.